This is Mr. Wartsky, and today we are going to start the first screencast for the nervous system. So let's get started, shall we? So the nervous system monitors both the outer and inner world. There are three functions of the nervous system: sensory input, we sense the sense; integration, we interpret the information, so basically we make sense of the sense; and the motor output, which are your muscles and glands, which respond to the sense. So we sense the sense, make sense of the sense, and respond to the sense. There are two types of nervous systems: the peripheral nervous system, which is responsible for your input and output, and the central nervous system, or the CNS, which is geared towards integration, making sense of the sense. And here's a picture of both the central nervous system, which is the brain and spinal cord, and the peripheral nervous system, which is everything else, including your senses. So there are two types of cells that make up the peripheral nervous system and the central nervous system. We have neurons and supporting cells. Neurons are also known as nerve cells, and you are going to draw a picture of a nerve cell, labeling the following: the dendrite, the cell body; the axon hillock; the axon, the myelin sheath; the nodes of Ranvier; the presynaptic bulb; the synapse; the postsynaptic dendrite. And that's it. Now, it's on the next slide, but I want to tell you that there are three types of neurons. There's a sensory neuron, which picks up the sense; the interneuron, which carries the messages back and forth and makes sense of the sense; and the motor neuron, which responds to the sense. So here we go. Here's a picture of the neuron. We have the dendrites, which receive the impulse from the other neuron. I want to write that down. It receives the impulse from the other neuron. The nucleus that contains the code, the DNA, you know that. The cell body is pretty much the cell which contains all of the organelles. The axon is this long thing that goes all the way down here. Now, the axon hillock is where the axon meets the cell body. The axon meets the cell body at the axon hillock. The myelin sheath is the covering that goes around the axon. Schwann cells are a type of cell that produces the myelin sheath. Type of cell that produces the myelin sheath, along with oligodendrocytes. You're going to learn about that in a moment. And the nodes of Ranvier are between the myelin sheath here. See, right there. And we have the axon terminal, the end of the axon, which will usually connect to another neuron or the gland or muscle. Draw that. Hit pause, draw it down. Let's go right down to the definitions. Get going. Hurry up. So the supporting cells, neuroglia, outnumber the neurons by 10 to 50 to 1. Now, they are essential for the structural integrity of the nervous system. In the central nervous system, the supporting cells are known as glial cells. What types of glial cells are there? Good question. We have astrocytes that their feet on their axons, their axon terminals, will encircle the capillary in the brain and it'll act as a blood-brain barrier. And certain drugs have to be able to pass the blood-brain barrier in order to be effective in working with the brain. Oligodendrocytes in the central nervous system for the myelin sheath, not the Schwann cells. Ependymal cells line the cavities that contain cerebral spinal fluid. Those cavities are called ventricles. And microglial cells are small cells that clean up cellular debris and trash and junk and protects against pathogens. Basically, they are the phagocytes of the central nervous system. And here are some pictures of glial cells. Neuroglials, we have ependymal cells. Microglia cleans up astrocytes. See these little things here? Those will encircle the capillaries. Oligodendrocytes form myelin sheath. The Schwann cell is the only part of the peripheral nervous system. See, peripheral nervous system. Beautiful. The peripheral nervous system, we have satellite cells, similar to they are in the, they're similar to the central nervous system. They're in the peripheral nervous system. They're on the back thing right here. Surround neurons, forms ganglia, regulate water. Ooh, we're going to regulate water. They're good. Oh, There you go. And Schwann cells, they form the myelin sheath. Now, all of these cells transmit an impulse. An impulse is an electric charge formed by the movement of ions. How do they move and what ions are moved? And those are excellent questions, which will be answered shortly. Here we go with glial cells. More, huh? There we go. So let's talk about the resting potential. This is a neuron at rest. So all neurons have a charge difference across the cell membrane, and we can actually measure the difference. The charge of a resting neuron is negative 70 millivolts. That's when we compare the inside of the neuron to the outside. Now, the outside is 0 millivolts. That's what we're going to use as our reference. So the inside of the neuron is more negative or less positive than the outside. Sorry about that. That was the phone. Took care of it. So the charge of the resting neuron is negative 70, right? So we compare that with the inside, the outside. The inside is more negative than the outside. So there are 4 different charged particles that lead to the difference in charge. We have sodium, potassium, chloride, and A minus. That's not your grade, although you probably hope for it. And A minus is a large protein, different proteins, and they all have negative charges. Now, the only thing that can't move out of the neuron is the A minus. So the ions move in and out of the neuron through channels. These channels select when the ions move and which ions move through the neuron membrane. Okay, these channels are very specific. Now, we said before that the A minus proteins, they never leave the neuron. So let's first talk about potassium, because potassium and sodium are the major ions that we're going to focus our attention on. So potassium will move through the potassium channels until an equilibrium is met, right? So you're not going to have all the potassium move in, because by diffusion they'll move in and out until they have an equal concentration. Now, some of the potassiums are attracted to the A minus. We got that. So if we measure the charge potential inside and outside, it's going to be negative 85 millivolts, and that's called an equilibrium potential. That's when the potassium reaches equilibrium. Sodium. Sodium will also move, but they'll move through sodium channels in and out, and then there's going to be more sodium moving in because of the negative A ions or A proteins inside, which is fine. So when all is said and done, when the sodium stops moving, then we're going to have a negative 70 millivolts. That is our resting potential, right? That's negative 70 millivolts. Now, there's a protein called the sodium potassium pump, and that uses ATP so that you know that it has to be active transport. And what will happen is it will move potassium inside the neuron and sodium outside the neuron. So the charge is going to be the same because we're moving the positive ions, but we're creating a concentration gradient, which means that there's going to be a higher concentration of potassium on the inside, a low concentration of potassium on the outside of the neuron, a high concentration of sodium on the outside of the neuron, and a low concentration of sodium on the inside of the neuron. Right? Now, the ions can't move because the channels are closed and they're keeping them from moving back and forth. But you know that the sodium wants to move inside, and you know that the potassium wants to leave the neuron. So here we go. We have the proteins minus here. We have the potassiums moving in and out, with the sodiums moving in and out, and then eventually the sodium-potassium pump with ATP is going to kick sodium out, potassium in. And over negative 70 millivolts. So an impulse is a change in charge. The ions are going to move, so that will change the charge. Right? So it changes it from a negative to a positive. I think I got that wrong right there. Negative to a positive, that's an action potential. So a stimulus will cause the sodium channels in an area to open, and sodium will rush in because of the concentration gradient. As sodium rushes in, the charge will increase because we're adding positive to a negative. So if that impulse hits a threshold, if enough sodium enters, and it hits about -50, which is about 15 to 20 higher than the resting potential, all the sodium channels in the area are going to open, and all the sodium is going to rush in, and all these positive charges are going to go into the neuron, and it's going to go from that -50, that threshold, all the way to +30. So if you look at it from the beginning, from -70 to +30, that's a 100 millivolt difference. This is called depolarization. Now, if you don't hit the threshold, if the impulse isn't strong enough to cause enough sodium channels to open, then it's going to be like nothing ever happened. You're not going to sense the sense. You're not going to know what ever happened. It's going to go right back to the resting potential because of the sodium potassium pump. The nerve impulse is called an all or nothing. Either you feel it or you don't. And there's the action potential with the sodium flying in. The potassium cannot leave. So we have an increase of 100 millivolts from all that sodium rushing in. All right, so all the sodium is rushing in here. Beautiful. And it's going to eventually, this area is going to cause this area to depolarize, which causes this area to depolarize, which causes this area to depolarize. So the depolarization moves down the axon like a wave. So once depolarized, we need to get back to resting potential, right? So how do you do that? Well, you need to get first back to the correct charge. So the potassium channel is open and all of the sodium rushes out. And this is called repolarization. Now, too many potassium will leave and the charge goes below -70 millivolts, and that's the undershoot. Now, some of the potassium will move back because the potassium channels are open, right? And then we have the -70 millivolts be established, and then we have to have the sodium potassium pump reestablish the concentration gradient with sodium and potassium. Don't forget to hit pause if you need to. And right here, now with repolarization, the sodium channels are closed, the potassium channels are open, the potassium flies out of there. And then the potassium can go back and forth here to reestablish the resting potential. So we have this graph, right? So here is our resting potential. This is our threshold, right? If the threshold is met, all the gates open up in that area. We go all the way up to +30. Sodium rushes in. Potassium rushes out right here. Too much potassium rushes out. And then some potassium moves back in, and we get the resting potential right there. Draw the graph, draw what happens, explain what happens in each step of the graph. You should be able to do that. The action potential moves in one area, causes the sodium channels of the next area to open. Right? So the action potential moves down the neuron like a wave in the stadium, but that is too slow. So the solution is saltatory conduction. Because we have the myelin sheaths around the axon, those sodium and potassium channels are blocked. The only time you have sodium and potassium channels are at the nodes of Ranvier. So when you have depolarization at one node of Ranvier, it causes the depolarization at the next node of Ranvier, which causes the depolarization at the next node of Ranvier. So the impulse basically skips from node to node to node. This will increase the rate of transmission incredibly. Now with MS, you demyelinate, so our impulses are slower. Right there. The depolarization happens from node to node to node. So the impulse will skip down the axon. So we are going to stop there at the synapse. If you have any questions, please read your textbook, watch the video over again, or ask me in class tomorrow. Have a good night.